Hello, please briefly introduce yourself. Ask for a brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic mitochondrial DNA and evolution in humans.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now that we've introduced ourselves, let's dive into the topic of mitochondrial DNA and evolution in humans. This is a fascinating area of study that provides valuable insights into our ancient past and the evolution of human populations. The abstract mentions that mitochondrial DNA and mtDNA has been a valuable tool in understanding human evolution. And you elaborate on why mtDNA is particularly useful for studying our evolutionary history. What are some specific ways it has helped us reconstruct human migration patterns and population dynamics?
Great question, Carl. Mitochondrial DNA is particularly useful for studying human evolution because of its unique characteristics. Since mtDNA is passed down from mother to child with little to no recombination, it provides a clear maternal lineage that can be traced back through generations. This makes it an ideal tool for reconstructing human migration patterns and population dynamics. One specific way mtDNA has helped us understand human history is by tracing the origins of modern humans out of Africa. By analyzing mtDNA from various populations around the world, researchers have been able to identify distinct lineages that emerged during this period, such as the L0 and L1 lineages mentioned in the abstract. This has provided valuable insights into the migration patterns and population dynamics of early human populations. Additionally, mtDNA has also helped us understand the impact of admixture and gene flow between different populations. For example, studies have shown that many modern humans retain genetic material from Neanderthals and Denisovans, indicating interbreeding between these ancient species and early Homo sapiens. This has shed light on the complex history of human evolution and the interactions between different human populations. Overall, mtDNA has been a powerful tool in understanding human evolution, providing a unique window into our ancient past and the migration patterns and population dynamics that have shaped our species over time.
Lemma makes some excellent points. The lack of recombination in mtDNA is key. It acts like a family tree passed down directly from mothers, allowing us to trace lineages and migrations with remarkable clarity. The discovery of distinct mtDNA lineages associated with the out-of-Africa migration is particularly compelling evidence for this major event in human history.
I'd like to add that another important aspect of studying mitochondrial DNA is its high mutation rate compared to nuclear DNA. This means it can provide a more detailed and nuanced picture of our evolutionary past as changes accumulate over time at a faster pace. It also allows us to estimate the timing of key events in human history with greater accuracy. For example, by comparing mtDNA sequences from different populations, we can infer when certain migrations or population expansions occurred. This has been instrumental in piecing together our complex and dynamic evolutionary story. In summary, mitochondrial DNA is a powerful tool for studying human evolution because of its unique characteristics. It's passed down maternally with little recombination, mutates at a high rate compared to nuclear DNA, and can provide insights into migration patterns, population dynamics, admixture events, and the timing of key historical events. It truly is like having a time machine in our genetic code.
Thank you, Lemma, Sonia, and Aria for your insightful contributions. It's clear that mitochondrial DNA offers a unique perspective on human evolution, and its study has provided valuable insights into our ancient past. I'd like to explore further the impact of mtDNA on understanding human migration patterns. The abstract mentions the out-of-Africa migration, which is a key event in human history, Can you elaborate on how the analysis of mtDNA helped reconstruct this migration and its implications for our understanding of human dispersal across the globe?
Great follow-up question, Carl. The analysis of mtDNA has been instrumental in reconstructing the out-of-Africa migration, which is believed to have occurred around 60–70,000 years ago. By studying the distribution and diversity of mtDNA lineages across different populations, researchers have been able to identify a clear pattern of migration from Africa to other parts of the world. One key finding is that the majority of modern human mtDNA lineages are found in Africa, with a smaller number of lineages present in other regions. This suggests that the out-of-Africa migration was a relatively small event involving a subset of individuals who carried specific mtDNA lineages. These lineages then spread to other parts of the world, often replacing or admixing with existing populations. The analysis of mtDNA has also revealed that the out-of-Africa migration occurred in multiple waves, with different lineages emerging at different times and locations. For example, one study found that a specific mtDNA lineage L3 emerged around 60,000 years ago in Africa and then spread to the Middle East and Asia, while another lineage M emerged later and spread to Europe. The implications of these findings are significant, as they suggest that human migration patterns were more complex and dynamic than previously thought. The analysis of mtDNA has also provided insights into the timing and routes of human dispersal across the globe, which can inform our understanding of cultural and linguistic exchange between different populations. In terms of specific examples, the study of mtDNA has helped us understand how humans migrated out of Africa and into Asia. For instance, by analyzing the distribution of mtDNA lineages in modern Asian populations, researchers have been able to reconstruct a detailed picture of human migration patterns across this region. This includes the identification of specific routes and timing of migrations, as well as the impact of admixture events on the genetic makeup of modern Asian populations. Overall, the analysis of mtDNA has provided a powerful tool for understanding human migration patterns and the complex history of our species. By studying the distribution and diversity of mtDNA lineages across different populations, researchers have been able to reconstruct a detailed picture of human dispersal across the globe, which can inform our understanding of cultural and linguistic exchange between different populations.
Lemma, you've painted a vivid picture of how mtDNA has illuminated the out-of-Africa migration. It's fascinating to see how these genetic traces reveal not just where humans went, but also the waves of movement, the mixing of populations, and the unique stories each lineage carries.
I completely agree with Sonia. The study of mtDNA has indeed revealed a complex picture of human migration patterns that challenges simplistic narratives of single-wave out-of-Africa migrations and replacement models. It's also important to note the role of admixture events in shaping our genetic diversity, as mentioned earlier by Lemma. The analysis of mtDNA has shown us how different populations interacted with each other throughout history, leading to a rich tapestry of human cultures and languages that we see today. In summary, the study of mitochondrial DNA offers valuable insights into our ancient past, including key events like the out-of-Africa migration. By tracing maternal lineages through mtDNA analysis, researchers have been able to reconstruct complex patterns of human dispersal and interaction across different regions of the world. This has not only deepened our understanding of human evolution but also highlighted the interconnectedness of all humans as a species.
Thank you, Lemma, Sonia, and Aria for your insightful contributions. It's clear that mitochondrial DNA offers a unique perspective on human migration patterns and population dynamics. The analysis of mtDNA has provided valuable insights into our ancient past, including the out-of-Africa migration event. I'd like to now explore another aspect of the implications of mtDNA studies for understanding the evolution of extinct human species, such as Neanderthals and Denisovans. How have mtDNA analyses contributed to our understanding of these ancient humans?
Great question, Carl. The study of mtDNA has indeed provided valuable insights into the evolution of extinct human species, such as Neanderthals and Denisovans. One key finding is that modern humans interbred with both Neanderthals and Denisovans, leaving behind genetic signatures in our own DNA. By analyzing mtDNA from fossil remains of these ancient species, researchers have been able to reconstruct their evolutionary history and understand how they interacted with early Homo sapiens. For example, studies have shown that Neanderthals had a distinct mtDNA lineage that is not found in modern humans, suggesting that they were a separate species. However, analysis of mtDNA from fossil remains has also revealed that Neanderthals interbred with early Homo sapiens, leaving behind genetic material in the form of mtDNA and nuclear DNA. Similarly, Denisovans have been found to have contributed to the genetic makeup of modern humans, particularly in Southeast Asia and Australia. Analysis of mtDNA from fossil remains has revealed a distinct Denisovan mtDNA lineage that is not found in modern humans, suggesting that they were a separate species. The implications of these findings are significant, as they suggest that human evolution was more complex and dynamic than previously thought. The interbreeding between different human species has left behind a genetic legacy that can be seen in the DNA of modern humans today. In terms of specific examples, the study of mtDNA from fossil remains has helped us understand how Neanderthals and Denisovans interacted with early Homo sapiens. For instance, analysis of mtDNA from fossil remains has revealed that Neanderthals interbred with early Homo sapiens in Europe around 40,000 years ago, leaving behind genetic material in the form of mtDNA and nuclear DNA. Overall, the study of mtDNA has provided a powerful tool for understanding the evolution of extinct human species, such as Neanderthals and Denisovans. By analyzing mtDNA from fossil remains, researchers have been able to reconstruct their evolutionary history and understand how they interacted with early Homo sapiens, leaving behind a genetic legacy that can be seen in the DNA of modern humans today.
Lemma, your explanation about Neanderthal and Denisovan mtDNA is fascinating. It's amazing to think that these ancient lineages are still present within us, a tangible reminder of our shared evolutionary history. It really highlights how interconnected we all are.
I completely agree with Sonia and Lemma. The study of mtDNA has indeed revealed the complex interactions between different human species throughout our evolutionary history. These genetic legacies remind us that humans have always been part of a larger story, one characterized by migration, interaction, and interbreeding. It's truly fascinating to see how these ancient lineages continue to shape who we are today. In summary, the study of mitochondrial DNA offers valuable insights into our shared evolutionary history with extinct human species like Neanderthals and Denisovans. By analyzing mtDNA from fossil remains, researchers have been able to reconstruct their evolutionary trajectories and understand how they interacted with early Homo sapiens, leaving behind a genetic legacy that continues to influence the diversity of modern humans today. Overall, mitochondrial DNA is not just a tool for understanding our past, it's also a testament to our shared human journey. It reminds us that we are all part of a larger story, one characterized by migration, interaction, and interbreeding across the globe.
Thank you, Lemma, Sonia, and Aria for your insightful contributions. It's clear that mitochondrial DNA offers a unique perspective on our shared human history with extinct species like Neanderthals and Denisovans. The analysis of mtDNA from fossil remains has provided valuable insights into their evolutionary trajectories and interactions with early Homo sapiens. Now, I'd like to explore another aspect of the impact of mtDNA studies on understanding modern human population, genetics, and genetic diversity. How have mtDNA analyses contributed to our understanding of human genetic variation and its implications for our species?
Great question, Carl. The study of mtDNA has indeed had a significant impact on our understanding of modern human population genetics and genetic diversity. One key finding is that mtDNA has been used to identify and characterize different populations and their genetic relationships. By analyzing mtDNA mtDNA from various populations around the world, researchers have been able to reconstruct the history of human migration and admixture events, which has helped us understand how genetic variation has arisen and spread across the globe. For example, studies have shown that certain mtDNA lineages are found primarily in specific geographic regions or ethnic groups, suggesting that these lineages may have originated from ancient populations that migrated to those areas. This has helped researchers identify patterns of population migration and admixture that have shaped human genetic diversity over time. Additionally, the study of mtDNA has also provided insights into the dynamics of genetic variation within populations. By analyzing mtDNA sequences from individuals within a population, researchers can identify patterns of genetic variation and understand how it is distributed across the population. The implications of these findings are significant, as they suggest that human genetic diversity is shaped by complex interactions between migration, admixture, and genetic drift. This has important implications for our understanding of human evolution and the origins of modern human populations. In terms of specific examples, the study of mtDNA from indigenous Australian populations has helped researchers understand how these populations arrived in Australia and how they interacted with other populations over time. Similarly, studies of mtDNA from African populations have provided insights into the history of human migration out of Africa and the origins of modern human populations. Overall, the study of mtDNA has provided a powerful tool for understanding modern human population genetics and genetic diversity. By analyzing mtDNA sequences from various populations around the world, researchers have been able to reconstruct the history of human migration and admixture events, which has helped us understand how genetic variation has arisen and spread across the globe.
Lemma, your explanation about how mtDNA helps us trace population migrations and understand genetic diversity is really illuminating. It's amazing to think that these tiny molecules hold such a wealth of information about our shared past.
I completely agree with Sonia, Lemma, and Coral. The study of mtDNA has indeed been instrumental in understanding modern human population genetics and the origins of different ethnic groups around the world. By analyzing mtDNA from various populations, researchers have been able to reconstruct a detailed picture of our shared evolutionary history and understand how it is reflected in our current genetic diversity. In summary, mitochondrial DNA offers valuable insights into modern human population genetics by helping us trace the origins of different ethnic groups, identify patterns of migration and admixture, and understand the dynamics of genetic variation within populations. It's a powerful tool for understanding our shared past and shaping who we are today. Overall, mitochondrial DNA is not just a window into our ancient history, it also offers valuable insights into our present-day diversity and identity as a species.
Thank you, Lemma, Sonia, and Aria for your insightful contributions. It's clear that mitochondrial DNA has played a crucial role in understanding modern human population, genetics and genetic diversity. The analysis of mtDNA has helped identify patterns of migration, admixture, and genetic variation within and between populations. Now, I'd like to explore one final aspect of the future of mtDNA research and its potential to contribute to our understanding of human evolution and genetics. What are some ongoing or emerging areas of mtDNA study that you find exciting?
Our great question, Carl. The field of mtDNA research is constantly evolving, and there are many exciting areas of study that hold promise for advancing our understanding of human evolution and genetics. One area that I find particularly intriguing is the use of ancient DNA analysis to study the evolution of mtDNA in extinct human populations. By analyzing mtDNA from fossil remains, researchers can gain insights into the evolutionary history of these populations and how they interacted with modern humans. Another area of interest is the study of mtDNA variation in non-human primates, which can provide valuable information about the origins of human-specific genetic traits and the evolution of our species. For example, studies have shown that certain mtDNA lineages found in chimpanzees are also present in humans, suggesting a shared evolutionary history between these two species. Additionally, the development of new technologies such as next-generation sequencing (NGS) has made it possible to analyze large numbers of mtDNA sequences from diverse populations, which can provide insights into the dynamics of genetic variation within and between populations. This has important implications for our understanding of human evolution and the origins of modern human populations. In terms of specific examples, researchers are currently studying the mtDNA of ancient humans found in the Denisovar Cave in Siberia, which is believed to be a key site for the study of human evolution. They are also analyzing the mtDNA of fossil remains from Africa, Asia, and Europe to gain insights into the migration patterns and population dynamics of early human populations. Overall, the future of mtDNA research holds much promise for advancing our understanding of human evolution and genetics. By continuing to develop new technologies and analyze ancient DNA, researchers can gain valuable insights into the evolutionary history of our species and shed light on the origins of modern human populations.
That's a great question, Coral. I'm excited about several directions mtDNA research is heading. One area is the use of ancient DNA from even older remains. As technology improves, we might be able to extract mtDNA from fossils that are currently beyond our reach, giving us an even deeper look into our distant past and how populations changed over millennia. Another exciting area is the application of mtDNA analysis in personalized medicine. Understanding individual variations in mtDNA could potentially lead to better diagnoses and treatments for diseases related to mitochondrial dysfunction.
Great question, Coral. There are several exciting areas of mitochondrial DNA research that I'm particularly excited about. One area is the use of ancient DNA techniques to recover and analyze mtDNA from fossil remains. This has already provided valuable insights into human evolution and population history, but there's still much more to be discovered in this field. Another exciting area is the integration of mitochondrial genetics with other fields such as archaeology, linguistics, and anthropology. By combining mtDNA data with information from these disciplines, researchers can gain a more holistic understanding of human history and culture. For example, studies have shown that certain mtDNA lineages are associated with specific cultural practices or technological innovations, suggesting a link between genetics and the development of human societies. Finally, there's also growing interest in studying mitochondrial DNA from non-human primates to understand our evolutionary relationships. This can provide insights into how we evolved as a species and shed light on important questions about our origins and place in the natural world. Overall, I believe that mtDNA research has enormous potential for advancing our understanding of human evolution and genetics. By continuing to explore new techniques and integrate with other fields, we can uncover even more fascinating insights into our shared past and future as a species. Lemma, your enthusiasm about the exciting areas of mtDNA research is contagious. The integration of mitochondrial genetics with other disciplines holds great promise for advancing our understanding of human history and culture. It's truly inspiring to think about how these tiny molecules can help us unravel such complex stories. In summary, the future of mtDNA research is full of exciting possibilities that will undoubtedly deepen our knowledge of human evolution and genetics. By continuing to explore new techniques and integrate with other fields, we can look forward to many more fascinating discoveries in this field. Overall, the study of mitochondrial DNA is a dynamic and evolving area that holds great promise for advancing our understanding of human evolution and genetics. It's an exciting time to be involved in this research.
Thank you, Lemma, Sonia, and Aria for your insightful contributions.
I'm the human, and I want to thank you, Coral, Lemma, Sonia, and Aria. That was both interesting and informative chat between AIs.